Everyone, welcome back to my channel Xgene and proteinX. In this tutorial video, I'm going to talk about the difference in between prokaryotic and eukaryotic DNA replication. So here, I'm not going to talk in detail on this DNA replication. I already made separated videos on prokaryotic and eukaryotic system. So if you haven't watched those videos, kindly find the link from the description box and watch those videos. Let's begin. In the prokaryotic system, it does not have any nucleus. That's what this replication it occurs in the cytosol. Whereas this DNA replication process it occurs in the nucleus in the eukaryotic system. And prokaryotic DNA is circular, and it has only single bubbles, not more than that. So as you see in this figure, so it has single bubbles and two fork, and this is DNA replication is bidirectional. Whereas in case of the eukaryotic system, the DNA is a long and linear DNA, and it has the multiple bubbles. As you see in this figure, there are multiple bubbles, and also there are a lot of fork. Number 3 is the for prokaryotic system, the DNA A recognize the origin of the replication and start initiation process, whereas in the eukaryotic system, and the ORC or origin recognition complex can recognize the origin of the replication sites and start initiation. So here the ORC can recognize, here the DNA A can recognize. Number 4 is the in prokaryotic origin of the replication site. It has two different kind of sequence, 9 Mars and 13 Mars. As you see in this figure, this is the 13 Mars and this is the 9 Mars sequence. But in the eukaryotic system, there is no such kind of sequence in the origin of the replication site. Number 5, that DNA B act as the DNA helicase, or it can unwind the DNA strand, and DNA B it can load by the DNA C loader in the prokaryotic system. But in case of the eukaryotic system, there is no such kind of DNA B or DNA C. So here the MCM complex it acts as the DNA helicase, so it unwind the DNA strand, and CDC6 and CDT1 it recruit the MCM complex. So next is the elongation process. So as we know that the elongation steps are carried out by the different DNA polymerase. So in the prokaryotic system, it has the DNA polymerase 1, it has the exonuclease and proofreading activity. So as we know that the exonuclease it can remove the RNA primers, and proofreading it can recognize the misbase pairing of the DNA. And whereas the DNA polymerase alpha it can initiate DNA replication, so it can only synthesize some short segment of the nucleotide. So DNA polymerase 2 it has the DNA repairing function, whereas the DNA polymerase epsilon it can synthesize the leading strand. So DNA polymerase 3 it can add nucleotide from 5' to 3' direction, whereas the DNA polymerase delta it synthesizes the lagging strand. So as you see in this here, that all of the DNA polymerase it can add nucleotide or it can replicate, but and also for the eukaryotic system, for leading strand and lagging strand it has some dedicated DNA polymerase. So for the leading strand it has DNA polymerase epsilon, and for a lagging strand it has the DNA polymerase delta. But in case of the prokaryotic system, the leading strand and lagging strand, both strand can synthesize by only the DNA polymerase 3. So out of those, only DNA polymerase 3 can as a function for the DNA replication. And in the prokaryotic system, it has only and there's 5 types of the DNA polymerase, whereas in the eukaryotes, it has 14 types of the DNA polymerase. So in the prokaryotes, the lagging strands are longer, is around 10 times longer, and it has the 2000 nucleotide base pair long, that lagging strand. But the eukaryotic lagging strands are shorter, it is 100 to 200 nucleotide long. and this DNA replication is fast, and this is 100 nucleotide per second it can replicate. But the eukaryotes, it is slow DNA replication. It can replicate only 50 to 100 nucleotide per second. And the prokaryotic DNA, it is the circular, so no end DNA. So there is no end DNA problem, end DNA replication problem. So there is no telomere and telomerase. Whereas in the eukaryotic, this is the long and there is a, it's not a circular, so it's a linear. So there is an end DNA replication problem, and the telomere and telomere is present. So prokaryotic DNA replication, it is very much simple, but eukaryotic DNA replication is much more complicated. So after replication, RNA primers cleaved and removed by the RNase H and DNA polymerase 1. But here in the eukaryotic system, after replication, RNA primers cleaved and removed by the RNase H as well as FAN1. But where here the DNA polymerase, it does not have any this kind of function. So let's have a look at the termination process. So in the prokaryotic system, the replication fork arrested by the TAR-TAS complex. So as you know that there in the terminus site, there are 10 different TAR-TAS complex, as you see in this figure. But in the eukaryotic system, there is no such kind of TAR-TAS complex. So it has the different kind of the mechanism to arrest the replication fork. So this is all about the difference in between prokaryotic and the eukaryotic DNA replication. So if you have any queries, kindly write in the comment section. I hope this video will be helpful. If you find so, kindly hit the like button and subscribe my channel. Thanks.